So I'm going to give a fast, because I don't want to disappoint Bjorn, energetic discussion of some new stuff that has just been funded in my lab. I would like to point out, although Eric isn't here, that this proposal was rejected by Beacon but accepted by the NIH. So I think you can draw your own inferences from that if you wish. As Bradley said, I'm also big about blogs. I'm also a big fan of open and online science, so I have a blog, and all the stuff I'm going to be talking about is available online in various ways. I think one of the remote connections got dropped. Maybe? Go ahead.
Hello. Just a sec. Renee.
Yeah. Renee, it's for you.
We'll call her. Well, she give it to Sophie.
So I'm particularly proud of the parts of my blog that are provide satires about the horror of the combination of data-intensive science and academia.
If that is you're capable, then that would be absolutely fabulous. All righty, thanks so much. Bye. The bridge just extended.
Oh, great, thanks. So anyway, okay. So I'm going to talk a little bit about the motivation for the work, then talk about reference-based versus reference-free approaches, and then describe what increasingly is my lab's sort of flagship research, which is single-pass algorithms, really efficient algorithms for doing lossy compression on resequencing data. So shotgun sequencing, I'm sure many of you have sat through far too many talks on this, but the basic idea is you're taking a pile of, say, books, like A Tale of Two Cities, many copies of A Tale of Two Cities, feeding them into a paper shredder, taking digitizing the words on the fragments, and then trying to put the books together just from the knowledge of what's on the fragments. So in this case, we have a bunch of short sentences, and your goal is to reconstruct the book, or the contents of the book, and do this for billions, potentially billions of fragments.
You don't have any errors in there. You could have put some errors in there.
So the trick here is that if this were if this digitization and shredding process were error-free, this would actually be quite easy. But in fact, what actually happens is that there are a lot of errors introduced in the sequencing process, in the reading process. And what you really want to do is use the fact that you have multiple copies of each of these sequences with random errors in them to reconstruct an error-free thing. And Luis, I've given you a taser, if you recall, before. If Chris asks more than 2 more questions, just yeah. So there's 3 sort of basic problems that people use next-gen sequencing for. There's resequencing, there's counting, and there's assembly. And I'm not going to talk about assembly much, although that's mostly what my lab's worked on. Assembly is a situation where you don't actually know the content of the book in the first place, and you're trying to put it back together. Resequencing and counting are where you sort of know the text of at least one version of the book, and you're trying to understand what the differences are between different editions of the book. So think seeing what's different between two people's genomes, or looking for, in this crowd, I guess I would imagine that evolutionary changes are particularly interesting. You're doing some sort of evolution experiment, and you want to know what's different where in the genome between the 2 lines. And this is now something we can do quite cheaply and easily. So the resequencing analysis problem is we have a reference genome, we know the Drosophila genome, or the E. coli genome, or the human genome, and we want to find the variants, which in this case are blue, in a background of errors, which are random and red in this case. So we know everything. We know the solid black line at the top, which is the reference genome. We have the black lines at the bottom, and we have a bunch of X's, and we want to know which of the X's are errors and which of the X's represent real variants, real differences. Counting is another problem, which is where you are doing things like mRNA-seq, where you want to do gene expression analysis. You want to count the number of times you observe a particular molecule based on reads being taken from that molecule. So the model that my lab started to use increasingly is this idea that next-gen sequencing is essentially making noisy observations of something, and your job is to extract the information from those noisy observations. And the information in this case is the correct sequence. And so when I talk about lossy compression, I didn't put the JPEG slides in here. When I talk about lossy compression, what I'm really talking about is taking a large data set that has a lot of noise in it and squeezing it down such that the information is preserved while the noise sort of goes away, trying to build generic techniques for doing this efficiently. So the real observation that we made, I guess, 3 years ago now, 2 years ago now, is that there's a lot of really redundant information. Really, a lot of the data that you get from next-gen sequencing is very redundant. That is, when you go in and you sequence books at random, you sequence genomes at random, generally you need to sequence on average to quite a high coverage. That is, you need to sample each sentence 10 to 100 times in order to be able to reconstruct the original full sentence. And that's because the sampling that you're doing is random. So if you look on the left here, we have, just for a simulation, you have position in genome, and then for an average 10x coverage, how many times you see that position in a read of length 100 bases. And the reason for the high variability there is you're reaching in at random and you're picking it out, and you're not doing it systematically. You're not systematically reaching in and picking out positions in the genome. You're just reaching in at random and pulling these things out. And so you get variation that looks basically Poisson or Gaussian based on the right in terms of the base coverage histogram that stems from this coverage diagram over on the left, which is sort of, in some cases you get hot, in some cases you oversample, and in some cases you undersample. And so whenever people talk about how you need 20x or 100x coverage for human genome resequencing, what they're really talking about is the reason you need that is because you want to minimize the number of times you don't see anything at all, right? The whole goal is to see everything at least enough times, 5 or 10 times, at least enough times that you can sort of say, I've seen it and I know what it is, and then error-free, wash out the errors by doing consensus calling across all 5 observations of that. So next-gen sequencing aims to shift this coverage histogram as far to the right as possible, because that means that there will be no things that have been seen 0 times, and everything will have been seen, say, 5 or 10 times on average. But the other observation, I think I have modified this here, the other observation is once you've seen something 10 times for assembly, or once you've seen something 20 or 50 times for resequencing analysis, there's no more information to be gained from that particular location. So if we can figure out how to efficiently go through this data and eliminate all of the stuff to the right of some average coverage, you can eliminate a lot of data without eliminating any particular information, and reduce the data set to the part of the data that you actually want and need to analyze, the part that contains the information you're after. And just to sort of motivate this, I was actually funded by the NIH for this, so I had to sprinkle the magic cancer words in the grant. I also have metagenomics, which is actually a much bigger problem, but people don't care as much about it at NIH. So if you think about where we're going with this, eventually, soon what we're going to be doing, people are already doing this, and soon this will be clinically feasible. We're going to be taking tumors, dissociating them into individual cells, and then sequencing, say, 1,000 randomly chosen cells from those tumors. This will be possible sort of at a commodity scale within 5 years. Which means that for each tumor cell that you sequence, you're getting between 60 and 300 gigabases of data. Sorry, the goal of this is to figure out population level information about the content of the tumor, to understand what the prime mutations were, and what the sort of what the causal mutations are likely to be, and what the correlative mutations, or just because it's a tumor cell, are. And you can do that by sort of doing phylogeny on lots and lots of tumor.
They're called driver and accessory mutations.
Viral and accessory.
No, driver
mutations. Sorry. And so the problem is that in order to do this, you generate, say, 60 to 300 gigabases of data for each cell. You have 1,000 or 1,000,000 tumor cells that you want to sequence. And that ends up being a lot of data, 60 to 300 terabases of data if you're doing this for 1,000 cells. And the current approaches to looking at variants are very computationally intensive and data heavy, in the sense that it's just really kind of unfeasible. You sort of have to start thinking about building big computing infrastructure for this, and it's not really a good idea to do that for sort of clinical and translational applications if you don't have to. So we're looking at doing this on things the size of a Raspberry Pi instead of on things the size of a HPC compute cluster. And so the current variant calling approach is you have a reference, you take all your reads, you map them to the reference, then you do what's called a pile-up, and you do variant calling on the pile-up. And you do downstream diagnostics on those variants. And just to give you this is a bad example, I just did some quick Google image searches. What you essentially are doing is you have some reference, you line up all the reads, and then you say, well, half the reads have an A and half the reads have a C. So there's a variant here that's a C, and it's a diploid organism. There's a variant here with a C, and there's a variant here with an A, so we call those variants. But this requires multiple passes across the data. First you line up all the data, then you look across all the data to see where there are changes. It's also got a number of problems. It's reference-based, so in particular. So there's a bunch of narrowly defined heuristics. You'll find that a lot of the current programs that take reads and map them to genomes are biased towards what human genomes look like, because that's what everybody wants to evaluate. That's what NIH-funded people want to do. They want to call variants in human and mouse. And so there are sort of guesses and heuristics and speed-ups that are put into these programs that are specifically for fairly low diversity human genomes. You also get something called allelic mapping bias, which is that when you're mapping everything to a reference, you tend to map things towards the reference allele rather than towards the variant allele. And that actually ends up skewing your results, and you need to apply corrections after the fact. And those corrections are generally empirical. You sort of train your variant column on that. It's also ignorant of unexpected novelty. So if you're looking at Craig Venture's genome is that of an older white male who's a little bit heavyweight. And there are a lot of people that are neither old, nor white, nor heavyweight, nor male. And so his genome may not be what you want to look at if you're looking for sort of novelty in terms of genomic islands or repeat structures or genes that might not be present in Craig Venture's genome. And so in general, this is a problem with these reference-based approaches. We know that we ignore indels, insertions and deletions. We know that we omit a lot of structural variation and a lot of true novelty. There's about a 0.1% difference between many human genomes. It's simply discarded because you're only looking at reads that map to something that you've already seen before. So if there's something there that you haven't seen, you're going to throw away that data. And then, of course, there's this other problem that the implementations that we currently have are multi-pass on big data. So just to give you two specific examples, this is a comparison by Terry Speed of TCGA variant callers on the Cancer Genome Atlas. And the details of this are a little bit tricky. They were actually doing variant calling on tumor samples. So they had normal, and then they had a tumor, they had somatic genome and then tumor genome. And they were looking to see how many of the variant caller pipelines, so this is the entire pipeline, gave you similar results. And what you can see, and I think I can say this is more or less their conclusion, is that the variant callers really suck. The pipelines really suck. When you look at 4 different variant callers, which are anonymized because they all suck, you find that 1,600 variants are called in common. And as soon as you say, well, what's called only in caller A and not in any others, you find 491 that are called by caller A. That's a lot. We're talking up to a 20% difference for some of these variant callers from one to the other. And these are due to differences in the heuristics they use to decide what's a real variant, what's not a real variant, what error models they've adopted, what they assume about the data. It's kind of bad. So progress needs to be made. And these things, I should say, these things are moving to the clinic, right? I mean, these are things that the Broad Institute does this on people's tumor samples to figure out how to treat them. So this isn't our little white tower academic, oh, this doesn't really matter to anybody. People will die or live because of this kind of thing. Hopefully not right now, but it's coming along, right? The other thing that we know, and this is from Zam Iqbal's work in Cortex, is that if most of the current aligners will only find places where there's an alignment that differs by a base. That is, there's some sequence, and we can map most of the sequence, and then there's a difference between, say, T and C, and that's called the substitution, a SNP, a single nucleotide polymorphism or variant. And here, we know that there are a lot of errors in genomes that are the results, or a lot of variants in genomes that are results of insertions or deletions of short nucleotide things, and if you look at the difference between the number of variants that are called with these indels, you see that the red line here, which is what everybody uses on the 1,000 Human Genome Project, is a considerable subset of what you can find if you use more sensitive techniques that are not based on mapping, that are based on De Bruijn-Graf techniques, actually, the black and blue. So you see many more variants outside the sort of narrow window that the mapping-based approach would call. So it's sort of a known situation, but the big problem right now is we lack a lot of tools to deal with this sort of insertion-deletion style variant calling. And so this is a great example of something I like to harp on, which is that the tools that we use obviously shape our ability to analyze the data, and most people are content with what the tools output and don't look any further than that, and that really limits our ability to understand what's really going on. Okay, so the challenges in this are that there's considerable amounts of noise in the data. Reference-based approaches have a lot of drawbacks, have a number of drawbacks, and they're what are primarily used. And the sort of way to summarize all of this is the first major data reduction step, the step where you take your reference genome, you take all your reads, and you boil all those reads down to there's a variant here or there isn't a variant here. It's extremely lossy in terms of potential information. Because that's a step where you lose all of this, and it can't be reconstructed by later steps. So from a computer science perspective, the question is, can we develop lossy compression approaches that reduce the data size and remove errors, and that retain all of the information? And I like to show a series of pictures here from JPEG where you can show that you can actually compress, you can do lossy compression on JPEGs where you dial down the quality of the JPEG quite a lot and dial down the file size of the JPEG quite a lot without really altering the image in the eyes of the human viewer. And so the sort of pipeline that I have envisioned is where you have raw data coming in, potentially even streaming off the sequencer. You feed it through this compression routine, which gets rid of 5 to 50, reduces the data in size 5 to 50 fold, that then can be fed into an analysis and potentially multiple analysis pipelines, at which point you can then deal with the information and feed it into downstream approaches. And at this point, if you can show that the compression retains all the potential information, that's all you have to store, and it makes life way better. And so the question, can we do this? And the answer is, we can for assembly, and I think we can for variant calling as well. And so the real idea is, it turns out that there are reasons why you need to save the raw data in cold storage. It's because if the doctor gets sued for the procedure, they need to be able to go back to the raw data. There's all sorts of very, very weird data retention policies and biomedical stuff that don't really scale when you're talking about hundreds of gigabases of data, but there you are. So you can go put this in cheap storage like Amazon Glacier, where they'll just charge you, a cent per gigabyte. And then you can keep the stuff that you may actually need to reanalyze as you develop better tools and better techniques and better analysis pipelines in much less expensive storage because it's so much smaller, but it'll be readily accessible storage. You can save that for reanalysis and investigation. So what we've been doing in my lab is operating across the spectrum of sort of developing novel theoretical advances in data structures and algorithms for dealing with this, implementing them at scale, which turns out to be a whole other problem, and then actually applying them to real data, which turns out to be yet another problem. Really our goal is to enable cloud computing-based analyses for everything. And I think just for sheer sexiness, I want to make this also work on a Raspberry Pi cluster. Because $30 cluster of $30 computers that can do this would be kind of just a neat toy, and they're $30, so I can buy plenty of them. I just fire a graduate student, and then okay. So don't look nervous, PP. So we've developed a variety of different things in my lab. Time and space efficient Kramer counting, which this is actually built on, which has already been published. In the general approach has already been published. We've applied it to Kramers, and QP's been working on a paper on that. And it lets us do things in less memory than anybody else. We've developed compressible assembly graphs where we can take all this data, look at all the connections between the data, and sort of compress it down again better than any possible exact approach. And that's actually been published in KBS. And then we've developed this online streaming lossy compression approach. And this is really a simple algorithm for taking essentially all of sequence analysis, all of next-gen, all of short-read sequence analysis, I should say, and moving it to a single-pass, low-memory algorithmic basis. And if you'd asked me 3 years ago, I would have given you all sorts of reasons why this was impossible. And that's why research is fun, because we sort of stumbled across this, and it's turned out to work quite well. So we can do all sorts of really great things with transcriptomes and microbial genomes and metagenomes in terms of assembling them de novo if we don't have a reference. And the question is, can we now apply this to variant calling? And so that's what I'll spend the rest of the next 5 to 10 minutes on. So the basic approach for this lossy compression is something called digital normalization. And digital normalization relies on this redundancy factoid, which is that as you collect reads, you can imagine that at some point you're collecting reads on this sort of unknown true sequence. You're doing this in the graph sense. So what you're doing is you get your first read and you say, okay, I have a read, and then you take your second read and you say, well, does this second read overlap in any way with the first read? And you get your next dozen reads and you ask, do these reads overlap with the reads I've already seen? And there's a fast and efficient ways of doing this that we've developed. And then eventually you get a read and you say, hey, I've already seen the region this read is from 5 times. I don't really need, or 10 times, or whatever parameter you want to set, I don't really need more than 5 reads for this particular region of the genome, because I've already seen it 5 times and I'm doing assembly or I'm doing variant calling, and I see It's a diploid, so if I collect 10 reads and I see no variance in 10 reads, then I can sort of guess that probably there's no variant here. Or you can do it for 20 if you want to be really statistically hardcore about things. And so at that point, you can now start throwing away the reads from the region that you've seen more. And essentially what you do is you make a local coverage estimate where you pile up all the reads. You make an estimate where you pile up all the reads and you say, okay, we've already seen this region enough times, now we can discard this thing. And the key point here is we can do this without knowing what the actual true reference sequence is, which is magic. It's actually really simple, but it's magic. And so what we end up with for digital normalization and assembly is that at the end of the day, we end up with a bunch of redundant reads. And we end up with a few reads that are the reads that contain the information. And these reads, the red reads, are all then redundant. And because the red reads also contain a bunch of random errors, we've also eliminated all the noise associated with that redundant information. And this reduces data sets by a factor of 80 to 95% for genome sequencing, and 99 to 99.5% for RNA sequencing because of the variable expression levels in terms of what's needed for assembly. So we can scale assembly by 1 to 3 orders of magnitude in terms of memory usage. But can we apply this to variants? And that's actually been, I guess, submitted but not published yet. So digital normalization here, it's a reference-free approach. It's single pass. It only looks at each read once. It doesn't collect the majority of the errors. It keeps all of the low coverage reads and retains essentially all of the information in the data set. And one of the funny things is it turns out it smooths out the coverage of regions, which helps assemblers do a better job of assembly, so we can actually get improved genome assemblies out of this in various circumstances. But this is about variant calling. Can we apply this approach to variant calling instead? And so the idea here is if we see a location that has a systematic set of variants in it, can we collect enough reads to cover both of those variants enough times to make statistical statements about those variants? And so this would be a single pass reference-free tunable streaming online variant calling mechanism, just to throw some buzzwords at it. And the short answer is yes, and this relies on some work done by my former PhD student, now Jason Pell, where he built a mechanism that lets us take reads and align them progressively to an assembly graph as we're building the assembly graph. And the assembly graph contains all of the information about variants that we have. So this is essentially a complete replacement for mapping. It's reference-free mapping, which it sounds oxymoronic, kind of is. And essentially it's a new technology. As far as we know, it hasn't been developed. This is novel with us, and it lets us very efficiently build pipelines like this. We take a read, we align the read to the graph, we build a model of the alignment that lets us decide whether or not something's likely to be an error or an insertion or deletion or whatever. We fix the read, so essentially we're doing error correction of the reads, and then we say, hey, now that we've error corrected this read, now that we've aligned this read as best possible in the context of all of the other reads that we've seen, does it still look like it has novelty? If it does, retain it. If it doesn't have novelty, saturated, just count it. Oh yeah, we've seen this portion of the graph n times, and discard the read. And at the point of saturation, we can actually call the variance. We can say, oh, we've got 20 reads here. 5 of them have an A, 15 of them have a T. Therefore, we can now call with some statistical confidence that there's a variant at this location that's an AT. And so essentially what we're doing, you can think of it a different way. This is really intuitively what's going on. Is we're adjusting the coverage that we retain as we go over the genome to retain the reads that have differences from the reference genome. So portions of the genome that just have a lot of low-level errors, most of the reads from those are discarded. Places where there are true variants are kept, and this actually does work, at least on simple data. Turns out repeats throw a little bit of a wrench into the mix. So we have to apply some heuristics there. And we're reasonably confident that we can make this work for human genomes, and we convinced reviewers at any rate, which I guess means we must be right now. I don't know how that works. But so essentially what we're working on is building this into a streaming and online algorithm. As the data comes off the sequencer, you could feed it base by base into this system, and even potentially stop sequencing as the variants become called with sufficient confidence. You can call the variants as the data emerges, so you don't have to wait for the data to come out and be finished and then run through things. It's independent of reference bias. We're adaptively adjusting the coverage to retain all of the signal. And there's an option for tuning. So the main question I get for this, well, yeah, but this isn't going to work for mixed populations, and that's correct up to a certain point. So if you know you have a diploid sample, you can say, I have a diploid sample, and I want to see, I want to retain 99.99% of all information, and we can tell you what parameter to tweak so that that is statistically guaranteed within the parameters of shotgun sequencing. If you have a population where you're looking for 1 in 10 alleles, you can't throw away as much data, you can't discard as much data as redundant, but you can guarantee that you collect all of those variants. This actually has real good, interesting applications for the human-associated microbes, where you're looking for strain variation, because we can discard all of the high abundance data and call variants on that while retaining all the low abundance data where the variant calling isn't going to work. So there's lots of sort of applications for this. And you can also do this neat thing where you can sort of subtract the reference. You can say, well, we have this human genome sample, we've already done variant calling on it, let's go back through and collect everything that our variant caller didn't pick up so we can reanalyze just that. So we're going to subtract the known reference and our variant called reference from all of this data in the process, and we can do that all in single pass without really any effort. And this is very similar to what Zam Iqbal's been doing with Cortex. And I think the main point that I like to make is everybody's like, well, yeah, but compute's kind of free, right? Like, why do you care? Well, it's kind of free until you start doing next-gen sequencing, and then it turns out to be more expensive than sequencing. And the main point that I want to make with that, though, is that the more compute you do later on, in terms of simulations, in terms of statistical calculations, in terms of looking at multiple samples, the better job you can do in terms of actually producing biologically relevant information. Right now, most people are wigged out about all the compute that's required to deal with the raw data, which is inherently kind of boring. And so I think that if we can move the compute bottlenecks to something more interesting later on, where we're resampling the data, or we're really doing an in-depth investigation of sequencing artifacts and errors, then that can be much more interesting than just dealing with this sort of torrent of raw data, where you really only want to look at the data once. And so that's sort of our model is, compute can be better moved can be better applied elsewhere and to do more intelligent things than just mapping and alignment, which inherently is kind of boring. Okay, so some concluding thoughts. We're trying to build both practical and theoretical leverage on this challenging problem. One of the neat things that I realized after talking to some supercomputing folk is that none of our current programming languages are going to work well on the architectures of the future. So increasingly, we're going to be moving towards low memory systems with lots of compute and not much data flow. And not much hard disk space, because it turns out building processors is still pretty cheap, but building all that I/O is really expensive. And all of our algorithms are many-core distributable, and so they can be used efficiently to pre-process the data before you go to a much smaller computer that can handle the data size. And we're also looking at how to move this into cloud or rental computing, so that institutions and clinics that don't necessarily want to have and maintain a supercomputing cluster on their on-premises can use rental computing as needed. Oh, I just sequence a bunch of stuff. Give me the answer. I'll pay the $100 for the compute. The other thing that I'd like to point out is that we actually build useful software. So a lot of our existing approaches are in use in dozens or hundreds of labs currently. And so we have every expectation of actually being able to turn this into a product that people can use, which is not always the goal of the funding agencies for some reason. There's some references in leading lists. I'll post this up on SlideShare. And I think I just want to end by thanking, this is primarily work that was enabled, this particular stuff is primarily work that was enabled by Jason Hell and Eric McDonald in my lab. And my funding was from the USDA primarily, and then some from the NSF and some from Beacon, and hopefully soon some officially from the NIH. So thanks, and I'll take any questions.
Have you looked into using graphics cards for parallel localization?
Yeah, so the big problem, that's like frequently asked question number 3. So the big problem is that the I/O for big data problems, for high throughput problems, the graphic cards don't have enough bandwidth to actually get the data on. The compute relative to the bandwidth is so cheap that that's not the bottleneck is getting the data onto the card, not doing the compute on it. But they're working hard on that. And there's a lot of really weird stuff going on with compute architectures now, where they're trying to sort of move everything close to one another on the chip and have GPUs. I don't really understand it in detail. and won't until I actually have to program it. But many core combined with GPU, combined with whatever else, and so then I'm sure some new bottleneck will emerge. But for right now, I think memory, we need a lot of memory for our current approaches. We need a lot of disk I/O access, and we're not dealing with many cores very well, or very efficiently. So it's a sort of a mess. Bioinformaticians don't develop good software in general. That's sort of how I'll just end that statement.
So you said it was reference free. Does that apply to indel?
Yeah.
Or just, I'm not understanding that. Because how would you know that you have an indel if you're not comparing it to your reference?
Right, so I guess what I mean by reference free is we're retaining reads and information independent of any knowledge reference. How you actually decide whether or not something's an indel may actually still be a tricky problem. XAMPP software, the Cortex actually will give you, it will assemble both things. De novo, it will say, here's the sequence, and here's an alternate version of the sequence that has an indel here. So you can think of it as reference-free is another way of saying we're just going to assemble everything. But we're going to give you all the variants in the process of assembly. And then if you know one of the true sequences, you can subtract it. So I'm only interested in things that are different from what we believe the sequence to be.
So what's the minimum amount of coverage you need for variant?
Well, for a diploid, I think the argument nowadays is in the 20 to 30x range.
3x.
But I feel like that's the wrong question, right? Because the one thing we're not going to be lacking in the future is sequencing depth.
Right, but in terms of this, like the slide you showed where you have the camera bubble and you're stacking the reads, counting where each variant is. If you have a low coverage region, how do you know that that's a variant versus low coverage?
Well, so we know what the coverage is.
Right.
Right? So we're waiting until the coverage saturates to call a variant. So we can say, hey, here's this region, we didn't get enough information to really tell you, you deal with it. Right? So it's actually a key part of the approach for assembly also is that we can basically tell you, yes, this region is done, you can assemble this now, versus we don't have enough information according to your specified parameters, so buyer beware in some sense. And I think that kind of statement to the user is something that a lot of bioinformatics software also doesn't currently make. Right? Here's the uncertainty in what we're telling you. Don't trust us. Right? It would be really nice to see, but that's sort of the thought process we're trying to bring to it.
So then the transcriptome assembly though, so if you've got, that's like almost combining 2 or 3 bundles of the same organism together. So how do you foresee that working?
Transcriptome assembly?
Yeah, with this.
You mean to do
RNA-seq.
allele-specific expression, or
Yeah.
to just do straight? Well, all the coverage is local. So I can tell you this transcript is 1000x, and we've saturated the alleles to 20x, and here's the 2 alleles. And we can say this transcript's 5, abundance 5, so we can't tell you the difference between the alleles and the errors for this. Sequence more.
Right.
Yeah. But, yeah. I think the main point is we're not going to do anything with information. We're trying to avoid losing any information. So inherently, by designing it that way, we're trying to guarantee that we're not throwing away anything interesting. That may mean we're retaining a bunch of stuff that isn't interesting in order to guarantee that we don't throw away too much stuff. But that's actually, you can still, it turns out, throw away 90% of your data as uninteresting with high confidence.
So would you be able to, like the indel question, if you don't have a reference and you're not sure what's in the reference, anything from microbial stuff to the cancer genome, how would you get around that? Just to give a low confidence score to those areas? Or?
I think the question. So I'm gonna cheat. The question is, what's your biology question?
Well,
Because what you really want to do at the end of the day is say, I'm really not interested in indels that are in non-coding regions, but I'm definitely interested in indels that are in exons.
Sure.
So here's the exons I'm interested in. So the goal is not to necessarily call with high confidence, this is an indel. The goal is to give all of the information necessary to decide that to downstream programs so they can do it in the context of whatever biology you're applying it to. So it's very much a pipeline thinking rather than an end user statement. Are there any questions from, I'm guessing, is that Idaho? Okay. No good, no, oh, no questions. I can't tell if this
I
is.
actually have a question.
Yeah, go ahead.
It's sort of in reference to what you were just saying about the context of these variants. Do you have any approach to figure out what the context of these variants is if you don't have a reference? I mean, it seems like it's something you still got to tie this back to the gene that it's in, or the exon it's in, or
Yeah. So that really goes back to the work that my lab's mostly been doing, which is on assembly. And so we're looking at ways to not just at the simplest, what you would do, the way you would currently do that is you'd say, I'm going to assemble my data, I'm going to take the assembled data, and I'm going to blast it against something, and I'm going to find the genes that are homologous to genes that I know something about, and I'm going to go back and look for variants in those genes. Or antibiotic resistance genes would be one example in human-associated communities, human-associated microbial communities. So that's what we would still do here. That's actually what the current variant callers, what the Cortex variant caller does, is it will let you say, I'm interested in this region because it looks like a gene that I know something about. We're actually trying to pull back to the point where we can go directly in at the graph level, the assembly graph level, and pull out genes based on either hidden Markov model matches, which we actually have working, or BLAST, or just open reading frame calling. But that's sort of building the whole software stack ourselves, which is going to take a little while. I think at the moment what we're going to be able to do is assemble something and then tell you what variants there are in the assembly at what abundance when you don't have a reference. And when you do have a reference, we will tie it into the reference that you do have and say, here's where the variant is according to the reference that you currently have. You can think of it as...
So that's going to mostly be...
Go ahead.
That's going to mostly be heterozygous variants. Like if you have a homozygous region and it's variant to some reference, then obviously you can't find it, right?
If you have, well, yeah, right. If you don't have, if you don't have a comparison. But I mean, I think, where we're going with this is soon we're going to have nothing but sequence information from sort of everything. And so I'm reasonably comfortable with that approach because we're not going to lack the ability to sequence everything we're interested in. We're going to lack the ability to extract sensitive variant calls from it instead. But we're trying to build a general purpose toolkit that will let us take whatever data you have and get as much out of it as possible. In many cases, if you don't know what you're looking for, which is increasingly the case, it's going to be hard to do. But we'll talk about soil metagenomics sometime. And that's the thing. All right, well, thank you very much, and I guess we're